Good afternoon, everybody. I'm Chip Schooley from the Division of Infectious Diseases and Global Public Health, and I'm delighted to have the pleasure of welcoming you to our IPATH seminar this afternoon, where we have a very special guest from the University of California, San Francisco, Dr. Jayant Rajan, who is a professor in the Division of Experimental Medicine and has a wealth of experience and a wealth of degrees that extend from his MD to an MPH to a PhD. And he's now settled into a very exciting series of studies that use phage as a way to explore serological responses, and is going to share some of that with us today. So I'm going to try to get control of the mouse so that we can. This mouse, yes, we have mice, not mouse today. There we go. So now I'll turn the floor over to Jay rather than spending half of his time introducing him, and leave more time for questions at the end. Jay, welcome. Thank you.
Thank you, Chip. Thanks for that introduction. It's great to be here. I think I'm actually going to take my mask off if everybody's okay with that, because I think my voice is otherwise going to get somewhat muffled. Great. Okay. Reset my timer here. Well, again, thanks for having me here today. It's a real pleasure to be in San Diego. I haven't been here in a little while, and my wife has never been here, so it's and the weather's fantastic. So I'm going to talk to you all today about the work I've been doing over the last few years, which is using bacteriophage as a tool to probe serological responses to infectious diseases. And I want to thank IPATH for having me down here. I know that the focus is very much on therapeutics, but there's lots of things we can do with phage. So I hope this isn't a controversial statement that antibodies are key mediators of host defense against infectious diseases. They bind toxins, they bind pathogens, they promote opsonization of invading pathogens, and they activate complement. And we all know that folks who have deficiencies in antibodies have increased susceptibility to infection. This is just a figure that I stole out of an old edition of Janeway, just to make a very basic point. So when we think, studying antibodies is important, and a lot can be learned about host defense, and we have a few tools for doing so. One of the classical tools are functional assays, so things such as neutralization assays. And these are incredibly important because they tell you something about what the antibody is actually doing, but they're a relatively low throughput. There's only so many things against which you can measure neutralization simultaneously. Another technique that we can use to probe humoral immunity is IG-seq, so immunoglobulin repertoire sequencing. This is a high throughput approach, but in the end, it doesn't necessarily tell you anything about antibody function, because you can't always infer function simply from antibody sequence. And then last but not least, there's what I do, which is phage or phage immunoprecipitation sequencing. And so this is kind of, I don't want to call it the best of both worlds, but it combines the high throughput nature of IG-seq with the addition of some functional characterization, in that what you do get are binding specificities of antibodies. So I know this is super basic for folks here who've been working on phage for a while, but just as a reminder, what bacteriophage are viruses that infect bacteria, and they have exquisite host specificity. And usually when a bacteriophage infects its host, there's one of two possible outcomes. One outcome is that the bacteriophage enters the cell, and injects its DNA in the cell, replicates, and results in lysis of the cell. And this is sort of the classic lytic cycle. And another outcome is that the bacterial genome either maintains itself as an episome or integrates into the host cell chromosome and is replicated with it, but doesn't actually result in host cell lysis. And just stepping back, I think I always like to remind folks that bacteriophage have been critically important in molecular biology. A lot of the enzymes we use in the lab, restriction enzymes, DNA ligases, T7 RNA polymerase, are proteins that derive from bacteriophage. And so a lot of the fundamental insights we gained about transcription and translation and so on back 40, 50 years ago were built on the back of bacteriophage. So there are lots of different bacteriophage out there. They come in all shapes and sizes. And just looking at this image that I stole from a relatively recent Nature Micro Reviews review article from Nature Reviews in Microbiology, one can see structural similarities with eukaryotic viruses. While there's lots of different bacteriophage out there, in the lab, two that are very frequently used are M13, which is a filamentous phage, and T7. And so one of the key features of bacteriophage is their genetic pliability. And the reason M13 and T7 have been such workhorses in the lab is that it's possible to engineer surface proteins in both so that you can express an exogenous antigen as a fusion protein. So for M13 in particular, this was leveraged for display of antibody fragments or Fabs and SCFVs, and has long been a workhorse for the development of monoclonal antibodies. So really important reagents in the lab. So important enough that in 2018, a Nobel Prize was awarded to Smith and Winter for quote unquote phage display of peptides and antibodies. So really fundamental stuff, really important biology, just a powerful tool that has been in use for decades and is continuing to get refined and used. So what I do is what I like to think of as phage display V2. It's something called phipseq, and which is short for phage immunoprecipitation sequencing. And what phipseq is essentially is the marriage of two robust technologies. It's the marriage of phage display with commercial DNA synthesis. And so what it really is a way to interrogate antibody responses against linear peptides. So the process for doing phipseq is relatively straightforward. One identifies a proteome of interest against which one is interested in looking at serological responses. So the starting point is typically NCBI. You download some sequences from RefSeq. The proteins in that proteome are then broken down into sets of overlapping peptides, and both the length of the peptides and the degree of overlap of the peptides are parameters that can be varied. One designs oligonucleotide sequences to code for these protein sequences, clones them in parallel into a phage display vector. So commonly for phipseq, what's used is T7, but there's no reason one couldn't also potentially do M13. And what you get in the end is a mixture of phage, each one expressing a different peptide on its surface, where you know what the genetic encoding is. And so that means that what you can do ultimately is amplicon sequencing after you've done selection of phage using an antibody-containing sample. I'll talk a little bit more about that, how the whole process works in a second. So in T7, gene 10 is the major capsid protein, and it's pretty amenable to introduction of an exogenous antigen, and that's basically what we end up doing in phipseq. So once you've made your phage library that has phage each expressing a different peptide, essentially what one does is an immunoprecipitation experiment. So starting with a sample that contains antibodies, one incubates that sample with a phage library. One captures the antibodies using protein A and G beads, so just doing an immunoprecipitation. Any bound phage are also captured, while any uncaptured phage are basically liberated. And then usually what we do is multiple rounds of selection with the idea of increasing specificity. And at the end, once one has done however much selection one wants, the readout is amplicon sequencing, and what you end up with is a table of peptide counts by every single sample that you've processed. So to give you an example of how this works in practice, so I work with Joe DeRisi at UCSF, and the DeRisi lab has been doing work on phipseq for a while now. And really where this all started for them was in autoimmune disease, and particularly neuroautoimmune disease. So this is the case of a 15-year-old girl who had been diagnosed years ago with anti-Hu antibody encephalitis and seizures. She had scarring in the hippocampus, as pointed out by the yellow arrow here, visible on her MRI.
got treated with steroids, and then got years of IVIG, and then more recently got Rituxan and Cellcept. She was treated by the Mayo Clinic, but got septic on that, and has subsequently been treated on azathioprine, so on Imuran, and is doing clinically well. And as a consequence of her Imuran therapy, she developed IBD, and so now has ulcerative colitis. So this is somebody with a known disease with incredibly high titers, anti-Hu antibody titers. And we were able to obtain a CSF sample from her and probe our human proteome FIPSeq library using her CSF. And what we found was basically that what was enriched were known targets of anti-Hu. So just emphasizing the fact that you can use this technique to identify known targets, and just and you can get, in cases like this where there's a blazing signal on a clinical test, you also get a blazing signal by FIPSeq. But it's not just that you identify the antigen, it's because you've done this sort of relative dense tiling across an entire protein, you can also identify specific regions of reactivity. So in this case, this is not that patient, this is several other patients. It's a mixture of CSF and serum. And what you can see is that using this approach, you can identify immunodominant regions, and more importantly, that you can actually distinguish different subsets of patients using this approach. So it's a really powerful technique. And ultimately, I'm an infectious diseases doc. I think the autoimmune disease is certainly very interesting, both clinically and scientifically, but it's not really what I do. And so when I came into the DeRisi lab, what I was really interested in was applying FIPSeq to studying humoral immunity and serological responses in the case of infections. And so now what I'm going to move on to is talking about some of the work that I've done. I'm going to basically give you three cases, three applications that I've used FIPSeq for. The first is a study of dengue virus 1 infection and using a rhesus macaque model. This is in collaboration with folks at Walter Reed. The second is some SARS-CoV-2 work, which unsurprisingly, like everybody, ended up doing some SARS-CoV-2 work, kind of hard to avoid in this day and age. And then the last case is some more preliminary data where I've taken what I've learned doing viral profiling and applied it to a disease that have always been very interested in, which is tuberculosis. So just a few comments on dengue virus. Anybody that's been to South America or to South Asia knows that dengue is a very big deal. It causes considerable morbidity. And in its most severe form can even cause death. So dengue is a flavivirus. It's transmitted by mosquito. And because of the amount of morbidity that it causes, it's long been a target for vaccination, for the development of vaccines. And in fact, there's one vaccine from Sanofi Pasteur that has been approved. And there's a couple others that are very close to being approved. One from Takeda and another from NIH, along with a number of other candidates that are being evaluated. There's been a lot of caution about developing dengue vaccines because one of the classic conundrums of dengue is that there are 4 different serotypes of the virus, and if you get serial infections with 2 different serotypes, that's the scenario in which, particularly in children, that you can get the severest form of the disease, which is dengue hemorrhagic fever, which can be fatal. So vaccination is a very different scenario than natural infection or inoculation by mosquitoes. So the question that we were really interested in attacking and understanding was, does the route of infection, does delivery by a mosquito result in a different immune profile than injection? And so we're lucky enough to find collaborators at Walter Reed who had done precisely the experiment that we would have liked to have done that you really could never do in people. Which is they basically infected rhesus macaques, an N of 10 per group, with dengue virus 1 by mosquito bite. So they literally put the virus in the mosquitoes and allowed it to replicate and then let those mosquitoes bite the rhesus macaques, and also by subcutaneous injection. And so
the key thing was that they followed the animals longitudinally. So we had baseline pre-vaccination samples, and then while they had quite a bit of sampling density, what we ended up settling on was looking at trying to capture early, mid, and late responses after the exposure to the pathogen. And so what I'm showing you here is the results of these experiments, and so what FIPSEQ data looks like. So on the top, in the top bar, what you see is every line is an individual rhesus macaque that was subcutaneously infected. And on the bottom line, you see every line is one of the rhesus macaques that was infected by a mosquito bite. And the x-axis is the entire dengue virus polyprotein. It's about 3,400 amino acids. So what you can see, it's 7 days post-infection. Unsurprisingly, in animals who are naive to dengue, you don't really see much of a response. By 35 days past post-infection, though, you see a pretty robust response. And what you're seeing is primarily a response in the envelope protein, which is the primary antigen against which antibodies are raised, and also the NS1 protein, which is also a known common antigen against which antibodies are raised. And both of these proteins play key roles in the pathogenesis of dengue virus. At 90 days post-infection, we see much the same thing, which is that there's continued reactivity in the subcutaneous group here, shown in blue, and in the mosquito group. There's substantial overlap between what we see in the subcutaneously infected and the mosquito group. And it's still mostly in the envelope protein and the NS1 protein. But at a year post-infection is where we started to notice some pretty significant differences. So while that initial big hump seen in the envelope protein is present and looks roughly the same between the subcutaneously infected and the mosquito-infected macaques, there are other regions in the envelope protein where there seems to be much higher reactivity in the mosquito-infected group. That's also true in the NS1, in several sites within the NS3 protein, and in several sites within the NS5 protein as well. So hopefully, looking at this, you can be convinced that there's a pretty substantial difference in how the immune profile evolves over time by the route of infection. FIPSEQ is very sensitive, and so it's really important to make sure that if you find something where you're interested in going after the biology, that you go the extra mile and validate that finding using an orthogonal method. And so I commented on the region in the envelope protein. It's in domain 3, here highlighted in gray. That was the region to me that seemed to be where there was the greatest difference. So I was able to validate that by ELISA. And what you can see here is that using a completely separate immunological assay, I basically got the same result, which is that the two groups look pretty similar at day 7, 35, and 90. But by a year post-infection, there's persistent reactivity in the mosquito-infected group that has pretty much returned to baseline or similar to day 7 in the subcutaneously infected group. The other thing I want to emphasize is that this approach has other benefits, is that you don't just get reactivity data. There's a limit to what you can put into a library, and that limit is really based on just how high the titer of a phage library you can get, and the degree of representation that you want of every single clone in your library. But I designed this library, which is a flavivirus focused library, to capture all of the genetic diversity that was present in all flaviviruses. And so basically when I designed this library in late 2017, I literally downloaded all of the sequences for dengue 1 and 2, 3, and 4 present in MCBI, and I did a bunch of bioinformatics. And essentially the take home point there is that you essentially can do mutational scanning using this approach. The other interesting thing that came out of this is that by using linear peptides displayed on phage, what we found was that there was considerable overlap in some of these antigens that we identified in particular this region in the domain 3 of the envelope protein, with reported MHC class 1 epitopes in from the IEDV. This is something that we've seen over and over again, suggesting that by doing these assays that we're actually getting additional, even more information than we realize. So in summary, what I hope I've shown you is that the route of infection for dengue virus really does have an impact on the duration of the response. And one question is what are the implications of this for the duration of vaccine-induced immunity? Again, injection with 4 strains of dengue at the same time is very different than serial infection by mosquito bite. And so it'll be interesting to see what the durability of vaccine responses is over time. In terms of where this work is likely to go in the future, this study focused on primary infection, but I think that our colleagues have also done some secondary infection experiments. You know, COVID has sort of made it difficult to get samples, but this is certainly another direction that we've been thinking about looking in. I think while I can tell you something about the antibody specificity using this approach, what really to take this to the next level, what we'd want to do is characterize antibodies. What are the functional characteristics? Are they neutralizing of the antibodies that bind some of these antigens that we're identifying, particularly those that are still present at late time points post-infection? And then last but not least, you know, these are studies that were done in macaques. And so one question is the critical question is, to what extent does this apply to humans? So comparing natural infection and vaccination in humans. This again is something that I've been working on, building relationships with collaborators in Brazil, where there's a large vaccine trial going on using the NIH vaccine conducted by the Instituto Butantan in São Paulo. So the bottom line is that using FIPSeq, we can identify differences in humoral immunity that I think would arguably be difficult to identify using other means. So this work, basically was finishing up towards the end of 2019 and the beginning of 2020. And essentially, the world changed, as we all know at that point. So in March of 2020, UCSF basically shut down all research, all in-person research, except for research that was SARS-CoV-2 focused. And having done a lot of development work on viral profiling, I was pretty well positioned to take a lot of what I learned and the tools that I built and apply it to SARS-CoV-2. And in particular, what we felt was that we had an excellent platform for biomarker discovery. At that time, in early 2020, there was a paucity of diagnostics, paucity of serological diagnostics for SARS-CoV-2. And so we thought, maybe we can help solve this problem. So as part of my prior work, I basically built an entire bioinformatic pipeline. It's just a bunch of R and Python scripts. And there are really 2 core functions that this pipeline had. The one was what I call the proteome processor, which is basically I'd written code that could take any proteome and just spit out oligonucleotides that I could then send to a commercial DNA synthesis vendor for sequencing. And the second is a data processing pipeline. So as I mentioned before, the output that one gets from this is sequencing data that needs to be pretty heavily processed for it to be understandable. And I've done some analytical method development as well, because understanding that generating the data is not easy, but it's relatively easy. Understanding the data is really where the challenge lies. So the thing I want to emphasize about this work is that it was really fast. So at the end of February 2020, I basically got the SARS-CoV-2 genome sequences available from NCBI. I downloaded it. By the beginning of March, I had basically crunched that through my pipeline and had designed oligos to code for SARS-CoV-2 peptides. By the second week of March, I'd sent off my order to Twist, which is the vendor that we used. I got back my oligos. I cloned them, grew up the library, sequenced it, did some initial validation experiments, and we were off to the races. At that point, I actually had to go on service in the hospital. And so the team that I was working with was able to run the samples. We were and so April was spent running the samples, analyzing the data, using an adapted version of my pipeline. And by the first week of May in 2020, we had identified what we thought was a good candidate biomarker set. So just want to walk you through sort of the steps of this entire process and how everything fully came together. So the first step was to basically obtain samples of people who'd been infected with SARS-CoV-2. And what we wanted to do was enrich for peptides that were found in persons with SARS-CoV-2, because these are the things that would be potential biomarkers for a diagnostic. And so what you're seeing here are what the profiles look for the set of positive controls that we had. And these are samples that we obtained from collaborators at UCSF as well at Rockefeller. And the profiling data is unsurprising for anybody who's been paying attention to SARS-CoV-2. What we mostly see is signal in the spike in the nucleoproteins. Interestingly, we'd incorporated some seasonal coronavirus peptides. There was very little cross-reactivity between seasonal and SARS-CoV-2. And we'd also previously built a pan-viral profiling library. And what you can see is that the results from the pan-virus library are similar to what we got from our SARS-CoV-2 specific library. Although in this case, what we mostly pulled down were bat coronavirus peptides, unsurprising since we know that that was the ultimate source of SARS-CoV-2. So once one does this enrichment of peptides using positive samples, what you want to do then is narrow down on things that might actually be useful in a serological diagnostic. And so what I'm showing you here is just a cartoon of how this process works. Is what we did was to basically do an enrichment using pooled human sera, take those enriched phage, plaque them, which what you're seeing on the far left. Then we basically counterstain those with known positive and negative, with known positive sera, identified plaques that lit up, and picked those blindly, not knowing what they were, what the specific peptides were. And after we did that, what we next did was to take those plaques and grow up pure phage from each one and print arrays, which we then probed using a set of positive and negative samples. And the point of doing this was to really try to, we had this unknown set of peptides that were enriched in people with SARS-CoV-2. And what we wanted to do was to find the smallest set that we could then potentially deploy as a serological diagnostic. So what I'm showing you here is just what the results of staining of one of these peptide arrays, which were printed using an acoustic liquid handler, the ECHO. And this is work that was really spearheaded by my co-first author on this paper, Colin Zemechnik, who's a bioengineer. And so he kind of came up with this idea of printing using an acoustic liquid handler. Once we've run a bunch of samples doing that, then essentially what we were able to do was figure out what the best set of peptides might be. And what we found was that we could, a set of 9 peptides could seem to have very good predictive power. So from a peptide pool of about 3,700 peptides, we were able to nail it down to 9 peptides. And at that point, it was sort of game on, right? Because that's tractable. Making a 9-valent serological diagnostic is something that is entirely feasible, as opposed to say using hundreds of antigens at the same time. So if you're wondering what these peptides look like, where did they map to? 8 of the 9 peptides map to the spike and the nucleoprotein, and these peptides are shown in as colored bars aligned across the genome, and with the intensity of basically the sort of the signal strength across all people shown above in red and blue, respectively, for spike and the nucleoprotein. The key take-home points here are that these peptides mapped in general to regions that were relatively highly conserved between SARS-CoV-2 and SARS-CoV-1. And then at the very bottom, what you can see is that not only were they highly conserved between SARS-CoV-2 and SARS-CoV-1, but there were also relatively short motifs that were conserved even across several bat coronaviruses. So just trying to dig into the biology a little bit more of what we were finding. So the final step was obviously the most important one, which was to take this set of antigens and deploy it as a diagnostic assay. And so to do so, now in theory, you could use printed arrays, right? Which is actually one of the huge strengths of this approach, because that's something you could do colorimetric assays pretty much anywhere. And so that was kind of, to us, one of the big selling points of this. So it's something that could be done in a low resource area. What we did was we took our phage displaying peptides, conjugated them to spectrally encoded beads, to Luminex beads, and implemented it as a Luminex assay. We then tested this on other samples that didn't go into sort of the generation of the peptide set, and had found that our performance was actually pretty good. Our sensitivity was 91%, and our specificity was 89%. And this was all done literally in a span of 2 months, and it was a pretty intense bit of work, but we thought that we had a great team, and we got it done. And it's work that we're very proud of. Of course, shortly thereafter, a glut of rapid diagnostic assays came out, and so we didn't really end up pushing this out into the world. We published the paper and made it available to anybody to use, but it's not something that we've pursued further. So my SARS-CoV-2 work actually continues because we built this library, and that has led to a lot of people seeking us out for collaboration. And one of the more interesting applications that I want to talk about very briefly is in looking at vaccination. So a colleague of mine in neurology at UCSF, Joe Sabatino, works on MS and collected a cohort of persons on different immunomodulatory therapies for MS. And we basically probed his samples using our FitSeq library. And what you can see is that some immunomodulatory therapies like ocrelizumab and rituximab, which are anti-B cell antibodies, essentially knock out the signal. And in folks without MS who are not on treatment, they have very similar responses despite having an autoimmune disease to people who are otherwise healthy. To me, the more interesting outcome of this work is the fact that we could actually detect differences between vaccination-induced immunity versus natural infection. So on the left, what you see is a summarization of profiles of people who were infected naturally. And on the right, what you see is folks who received mRNA vaccines. And to me, the interesting thing here is that in the mRNA vaccinated group, apart from the fact that the overall response and in terms of the number of people that mount a response seems to be more robust, it's also the fact that you get a much stronger signal in the N-terminal domain of the spike protein, which is a known target of neutralizing antibodies, than you do in natural infection. So using linear peptides to probe serological responses to SARS-CoV-2, we're essentially able to find what I would describe as a vaccination signal. So this is something that I'm very interested in pursuing further and am actively working on. So in summary, to talk a little bit more about, just to summarize what we did on SARS-CoV-2, really the key take-home point is that this is a proof of concept for a single workflow for rapid serologic test diagnostic development. And I think the one thing I can't hammer home enough is that what phage it ultimately is a limitless source and low-cost source of protein. And protein production, as anybody knows, can get pricey. To do this, all you need is a genomic sequence. And so while we apply this to SARS-CoV-2, it's in fact, the overall approach is applicable to many pathogens. So it's something that we are pretty bullish about in terms of being a platform for serological diagnostic development. And then last but not least, I just want to bring home again that we are able to actually measure differences between natural and vaccine-induced immunity. And so in short, I think that to me, what that tells me is that data that generated using this approach is both actionable and immunologically meaningful. Now I'm going to move on to the third use case and more recent work that I've been doing. And this work is more preliminary, but basically having learned a lot doing antiviral profiling, What I wanted to shift to was looking at tuberculosis. This is a disease that I've cared about for a long time. And I have some excellent collaborators at UCSF, and an interesting opportunity came up. So just as a reminder, TB is actually a huge deal in the world, right? It's the #1 infectious diseases related cause of death, you know, leaving aside the pandemic globally, and that's been the case for some time. It's even higher on the list between than HIV. Obviously, everybody, as we all know, there's a tremendous interaction between HIV and TB. TB is really difficult to diagnose. It can present in many different ways. And one of the big problems with TB is that screening is in large part driven by clinical symptoms such as cough. And, you know, while we do have diagnostic assays, some of which are relatively rapid, such as an AFP stain or Gene Expert, which is essentially PCR, the gold standard for diagnosis of TB remains culture, and it's slow. So you're talking 3 to 4 weeks in many cases before growth is even seen. And what this delay in diagnosis means is that in endemic settings, people are started where there's a high risk of TB exposure. People are started on treatment empirically. And obviously, if the culture ends up being negative, that treatment gets stopped. But, you know, you're talking about 4 drugs that somebody maybe does not necessarily need to take. One of which, rifampin, is infamous for interacting with other medications. So TB treatment, while relatively well tolerated, starting somebody on treatment is not trivial. Serology for TB is something that I would argue has largely been written off, and there are a lot of reasons for this. Part of it is the biology. I think the classic dogma of TB immunology is that T cells are more important than B cells. But that hasn't prevented people from coming up with serologic assays, and there are currently a ton of serological diagnostic assays that are used primarily in endemic settings, none of which have good performance characteristics when you're looking at sensitivity and specificity. People have done more high throughput looks at serological responses to TB. These have mainly been done using protein arrays where the proteins are expressed in either E. coli or yeast. There are not a lot of proteome-wide studies, and FIPSEQ, more importantly, is not something that's been applied to this problem. So having learned everything that I did, I thought, and I was able to sell my TB colleagues at UCSF on, that this was a really good use case for the technology. So my colleague, Jitya Kadabanchi, who's a pulmonologist at UCSF, had been involved with a cohort years ago called the MIND cohort, which is basically a prospective cohort study of inpatients with suspected TB. It was all adults with lower respiratory tract infections and cough of any duration. The key thing was that a sputum culture was obtained in all patients at baseline. So you really had ground truth for whether they had TB or not. So in the quest to find the triage diagnostic for TB, a serologic triage diagnostic, this was a really excellent population to start looking at. So going through the -80, we were able to come up with 168 samples from the original study, 39 of which we excluded because they were adjudicated as being unlikely TB, and one of which was excluded because their HIV status was unknown. This left us with 129 samples of patients with and without TB. And again, this is all based on culture, smear, and Gene Expert results in terms of adjudicating whether people had TB or not. The majority of the patients in this cohort were HIV positive. And among the HIV positive patients, unsurprisingly, the vast majority were smear negative. And among the HIV negative population, the majority were smear positive. So this is the main point I want to drive home about this, is that I ran all of these samples using FIPSEQ. I generated peptide profiles. I determined which peptides were enriched in each patient. And then I just did some simple hierarchical clustering. And what you can see is that without even trying very hard, I can pretty effectively separate pulmonary TB from people without TB, just on the basis of serological signatures, which is pretty exciting. So I showed this to my colleagues and they got super excited. And so the next step, and this work again is still ongoing, was to just do some machine learning. So I took the full sample data set and divided it into a training sample of about 90 samples, a training data of 91, and validation data that would not go into the training of 38, and focused on using regularized linear regression-based approaches to basically identify the best candidate biomarkers. I like these approaches because they're actually very interpretable, as opposed to sort of ensemble machine learning, where it's really a little bit more of a black box. And essentially, I did some cross-validation to figure out what the best modeling approach was, which turned out to be ElasticNet. Then I went back and I fit the model on the training, then on the entire training data, and tested its performance on the validation data. And what you can see is one of the key things with these methods is what you actually get is a series of models. So the selection of the model is really based on looking at, so for Lasso and ElasticNet, is there at least one model across the series of models, across every fold that performs well? And performs well in this case had an AUC of at least 0.7. And by that metric, the top dotted red line is an AUC of 0.7. The ElasticNet for most folds either got close to or was at 0.7, as opposed to the Lasso, which was a little bit more variable in its performance. So, when I went back and just made an ROC curve, I got an AUC of 0.67. But the thing that I want to emphasize is that I was able to narrow down from an initial set of 4,700 peptides to just 9. And this is really using a very small data set. Usually for machine learning, you're using much larger data set. This is with a training sample set of 91. So in summary, I think that we're pretty excited about this because it suggests that peptide signals are a promising target for developing a triage diagnostic. And what we're trying to do right now is just increase our N, because I think that that's going to be necessary to really tune our performance and make it better. So stay tuned. So in conclusion, what I hope you take away from my talk today is that FIPSeq is a very powerful platform for interrogating serological responses to infectious diseases. It has a lot of potential applications. So I talked about serological diagnostic development. It can also be used for vaccine antigen discovery, potentially for treatment response monitoring. It's really we just need to try some of these things out. There's also opportunities for further development of the technology to augment it so that maybe you get some functional characterization of antibodies. And maybe there's a way for us to adapt it so that we actually can measure antibodies against nonlinear and conformational epitopes. And in my own world, where these things are going is really in a vaccine direction. So I have a study that I'm conducting for. We showed this TB data to Gates, and they liked it. So I'm involved in a vaccine antigen discovery project through Gates. Also in discussions to do some antigen discovery for syphilis as well. And I continue to work on development of the technology. But what I want to close with is just reminding, just bringing home the message that I started with right off the bat, is that this is just one application. Bacteriophage can do so much more, right? There's a lot of work that's being done here on therapeutic applications. More work to be done to figure out how to really make it work as best as possible. But phage are a fundamental tool for studying protein evolution. I think the development of monoclonal antibodies is case one for that. It's a platform for understanding protein-protein interactions. It's a nanomaterial, potentially a drug delivery platform. And really, I think the limit is just up to what we can imagine and coming up with clever new ways to leverage this platform. And frankly, with some of the work that's going on here, where you guys are identifying other potential phage, we're working largely with M13 and T7. Who knows what else we could work with and what other cool things we can do with other phage that we can engineer. So I want to close by just a couple of acknowledgements. So all of this work was done with the support of Joe DeRisi, who's been a fantastic mentor and scientific collaborator. I can't say enough about how fun it's been to work together. It also, other person I want to specifically single out is Hannah Ritalik. Hannah Ritalik was an MD PhD student in the Derisi lab. You know, I'd sort of, some of you guys know my story a little bit. I'd walked away, I was a basic science guy. I walked away a little bit. Hannah Ritalik is the reason that I'm back at it. And Hannah and Joe made science fun for me again. And I can't thank them enough because I'm doing exactly what I always wanted to do. I'm doing what I did when I went to medical school, and it's been a blast. Michael Wilson has been a close collaborator. Callie Mandelbrem, Colin Zemechnik, Madura, and Elza are all folks that are part of our phage team and folks that I talk with a lot. I want to shout out to my TV collaborators. And then while I didn't really talk about any of the work I do in Brazil, I do want to acknowledge a couple of great collaborators that I have in Salvador and Sao Paulo. And that's a picture from Kings Canyon. So I'm a backpacker hiker and used to be an alpine climber when I lived in Seattle, hoping to get back into that. And that's a picture of my daughter Gaia when she was about a year old. She's now almost 2. Thank you. And
three areas that really show a lot of diversity in both approach and we have a couple of questions I think in the chat. There's one I think from Looney.
he asked a question, but then he
answered it. No?
had to leave.
Had to leave. Well, that will be a mystery. All right.
Okay.
Those of you who are outside and want to send questions in, please do so via the chat. Anyone have questions from here? Stephanie.
Yeah. I think, you know, obviously for any of the, for what I do, I think right now, in terms of the diagnostic development, I think it fits in sort of in very much the kind of case that I talked about in terms of identifying what the best potential predictors are. I think a lot depends on what the data one has is, right? I think that there are ways one could imagine using machine learning to inform protein design. Maybe it could be a useful tool for sort of figuring out how to engineer phage, kind of what we were talking about earlier today. How do you take T7 and make it infect Staph aureus, and stuff like that. But, it's a very powerful tool, but it really, it's a function that what you put in, it's the quality of the data that you feed into your machine learning is what determines what you're going to get out of it. But that would be like one obvious way that I could think where it can be used. You can think of doing experiments in the lab, where you basically are interrogating. You do a series of experiments with different phage, it's different pathogens that you're interested in infecting. Then you can maybe use machine learning to figure out, okay, what are the differences in the surface structural proteins that maybe track with why, phage A infects Staph aureus and phage B doesn't, would be one definite application.
Others in the room here? One of the things that is a barrier to therapeutics is having to use phages in the way they grew up in the world is finding the right pair. How much of a heavy lift do you think would be to have enough of a informatics platform on both sides of the bacteriophage pairing to be able to use that as a very strong predictor of which phages are going to be active against which bacteria.
Yeah, I mean, I think you'd need a pretty
Yeah.
big data set.
Yeah.
You know, and I think the other thing there is that probably what you'd want to feed into it is some structural information.
Yeah.
You know, because I think ultimately it's protein structure that determines
Yeah.
specificity. And so, you know, but those are solvable problems.
Yeah,
You
yeah.
know, and you know, I'm gonna hedge a little bit here, which is to say that if you had, you know, a slower dimensional but high quality structural data set,
Mhm.
maybe you could get away with less input.
Yeah. So you bring structural biology into the empiric data that you generate.
I think so. I think so, because I think that if the goal is to be able to design new phage or that you kind of want that structural information. That's probably important. Maybe you could solve it by just brute forcing it.
Yeah.
Right. And just if you screen thousands of different phage against, you know, a relatively small set of bacteria, maybe that could work. I mean, another way to do it would be to just do mutational,
Mhm.
right? And just randomly mutate capsid proteins or filament proteins and generate a library of phage variants that way.
Mhm.
And then just see what sticks and what doesn't.
Reassemble them. Yeah,
Exactly.
Yeah. Very good point. All right. Others? All right. Seeing none, thank you, everybody, for coming, and both in person and electronically. And thanks, Jay, particularly for coming in person. And Lizzie for organizing
Yeah.
this. That's right.
Thanks,
And your
Lizzie.
visit as well. So thanks, Lizzie, for everything, making sure that we don't end up with egg on our face as often as we would otherwise. So, all right. Thanks, everybody. Have a good rest of the day. Good weekend.